for example, the microgrooves that you can see in the image here were found to align cells along their axis. And this was quite useful from the perspective of trying to get cells to form different cell types, for example, growing neurons in aligned structures. And this collaboration, we think, started in the allotments because they were neighbors, and they both had neighboring allotments, but they produced quite a lot of research over 2 or 3 decades in this area. This type of collaboration led to a huge amount of research that even to this day, looking at how cells interact with topographies of different shapes and sizes, from the micro to the nanoscale. We have examples here of grooves that have been etched into surfaces and how cells align along these grooves, or how other cell types can use these nano islands as stepping stones, where the cell can adhere to these circular shapes on the surface. If we have pliable surfaces made of nano pillars, cells can actually bend them to their will. So cells actually pull on surfaces, and this is a good example of how cells can deform the materials around them through traction forces. Around the same time, there was this sort of awareness that cells are actually quite dynamic. And in this short video here, you can see how cell membrane actually ripples and ruffles at the nanoscale and at quite high speeds actually. And what we're actually seeing here is the membrane reaching out into the neighboring space around these cells, and the white specks that you can see are actually cell adhesions. You can see new adhesions forming where the membrane has probed the surface. So what's actually happening at these adhesion sites is the formation of groups of proteins. For example, integrins are these red proteins here, which then nucleates the formation of focal adhesions, which are collections of a variety of proteins listed here, and these connect onto the cell's cytoskeleton, which is a tensional structure which holds the cell in place and enables the cell to move. Interestingly, both the membrane motion and these integrin length sizes are at the nanoscale, and this was something that Professor Adam Curtis was quite interested in. So he saw these results coming from other groups, and he asked, can we interfere with how cells adhere to surfaces by moving the surface? Can we vibrate the surface, perhaps at the amplitude of a similar length scale to the cell integrins? And he came to the physics department at the University of Glasgow and asked for some help with this. And it was my PhD supervisor, Stuart Reid, who answered the call. And he, at the time, was in gravitational wave research at the University of Glasgow, and now he's in biomedical engineering at Strathclyde. But he helped Adam set up initial experiments vibrating Petri dishes with nanometer size amplitude vibration. And this started the research into the vibration of cells of a variety of types. So what we call nanokicking is actually nano-amplitude vibration. We're talking about having adherent cells growing on Petri dishes or other surfaces. And we're vibrating the Petri dish or the surface by roughly 30 nanometers. And we've taken a lot of effort to try and control this vibration so that we can understand what the cell is experiencing. So we can do some rough calculations, looking at the cell surface area, the mass of fluid that the cell is growing in. We can roughly estimate the sort of forces that the cell are experiencing to be in the piconewton range, and the peak forces are exerted during the peaks of the vibration. So during this, during the vibration, the cells are experiencing an accelerative force, and this basically stimulates the cell in ways I'll go over in a little bit later. So you might ask what this has to do with gravitational wave research. Well, to produce nanoscale amplitude vibration is non-trivial, particularly in complex materials such as Petri dishes and other containers. So we borrowed a few materials and methods from gravitational wave group at Glasgow, for example, so that we can do this effectively and reproducibly. The first piece of technology was a method of producing vibration, and here we've used piezo actuators attached to Petri dishes, and these are quite high accuracy, nanometer accuracy piezo actuators, sort that you might see in various interferometer systems, and obviously we need to control these with various control systems to produce the vibration frequencies and amplitudes we're interested in. The other technique is a way of measuring these 30 nanometer amplitudes. We need to be able to calibrate our Petri dishes. And here you can see a clip of a standard laser interferometer that you would use in a large ground-based or space-based gravitational wave detector, where you have the reflection along two beam arms of a laser, which is then passed through to a detector, where you can see fringes of interference if the length of these two arms changes. So if we imagine that our Petri dish is at one end of this arm, we're able to detect quite accurately motions in the nanometer range using the output interference of the changes in these two beam arms. In reality, this is the on the right here, you can see the actual interferometer that we use, which is a lot smaller than the ground-based detectors like LIGO, but is very sufficient enough to detect down to sub-nanometer resolution Petri dishes which we're vibrating. The last technique that we've been using quite extensively is finite element modeling, and this is quite a common sort of analysis that is seen in gravitational wave research, particularly around mirrors and their suspension wires. So this is a used silica cantilever that's been modeled in ANSYS in modal analysis. And you can see that we've sort of borrowed this sort of technique to look at our Petri dishes to try and understand which frequencies will excite resonance, so that we can understand what the cells are going to be experiencing if they're growing in one of these surfaces. Are we going to have a consistent stimulation of the cells, or are we going to be giving them a variety of stimuluses at different amplitudes, so that we can keep it as consistent as possible and we can really try and pin down what the cells are responding to. So the first apparatus that we sort of developed, well, Stuart developed, was using piezo actuators to vibrate Petri dishes. And since then, we've sort of scaled this up, looking at culture plates and then flasks, using these multi-piezo actuator arrays. And more recently, we've gone on to vibrating even larger cell culture vessels in these multi-layer cell stacks. And this allows us to go from vibrating a few thousand cells up to tens of millions, the focus being on delivering enough cells for clinical use. So you can see over the past 9 or so years how we've sequentially been upgrading the devices we're using, moving towards something a bit more industrially relevant. So just looking at the Petri dishes as an example, here are some ANSYS modal analysis, just showing how I think these are the first two modal frequencies of the Petri dishes at A and B, and you can see that there is a lot of distortion of the dishes, particularly in B. You can see that the outside edge of the Petri dish, the cells are going to experience a lot more vibration forces, whereas what we've done in image C and D is reinforce the dish using an aluminium disc, and this smooths out the vibration amplitude across the cell growth area. We still get the deformation of the sides of the dish, and that could be interesting in terms of fluid motion, which could impact the cells as well. But in terms of the vibration itself, we believe that we're delivering a consistent vibration amplitude based on these models. Moving on to the sort of larger devices that you saw in the pictures before, we've also used laser interferometry, in this case scanning laser interferometry, to measure the vibration patterns of these larger vibrating surfaces. Some of our first prototypes, we were just using glue basically to attach the actuators to the top plate. And you can see that we ended up with a sort of modal pattern, but it has high points and low points, which suggests that the consistency of these early devices wasn't quite as good as we wanted. And then my colleague Paul Campsey at Strathclyde came along and altered the design so that he could achieve this piston-like motion for the vibration device. And these are amplitudes in the range of 30 nanometers. We're quite good now at controlling specifically down to around 5 nanometers how accurate across the surface of one of these devices the vibration is. And that just means that we can really tell what we're doing to the cells. We can do comparative experiments so that we can look at 20 versus 60 nanometers or even higher amplitudes to see how the cells. We've done a lot of analysis of the cultureware, vibrational analysis. This is harmonic response modeling done in ANSYS on the surfaces of a 6-well cell culture plate. And you can see that the surface responds quite closely to the input vibration up to around 3,500 hertz, above which we start to get distortion of the culture plate. The image is actually at 1,000 hertz because that's a frequency we've been quite focused on in our cell culture work. But what this sort of shows is that we have a range of frequencies where we can quite closely control what cells are experiencing. We go above a certain range, then we start to lose that control. And we have to consider that we have less certainty of what the cells are experiencing. We've backed up the modeling with laser interferometry again, and we've used laser vibrometry to measure this at the cell culture surface using these attached pieces of silica, and you can see that the response with input voltage, we can very easily control the amplitude of the vibration. If we do a frequency sweep and we look at how the cultureware is responding to different frequencies, this kind of matches with our modeling, showing that above a certain point, around 2,500 hertz here, we start to see distortion between the center and the edges of each of these wells. We've also gone one step further and started looking at soft materials, because sometimes it's useful to grow cells in 3D hydrogels. So in these wells here are collagen gels, which we would grow cells suspended within. And using a similar technique with the laser vibrometry, we've measured again the response in terms of input voltage, showing that it's a linear response, and also I've done another frequency sweep to show that there's fairly accurate reproduction of the vibration at these lower frequencies. So what this has shown for us is that we can quite accurately produce vibration up to around 2000 hertz with this device, above which it gets a little bit less accurate. What you're seeing here actually is in the solid lines are the gel itself, whereas the dotted lines are the edge of the gel. So we can see that there is actually some distortion even at these frequencies where the edge of the gel is vibrating more or at a higher amplitude than the center of the gel. And the gel itself is slightly higher amplitude than the dish. So the soft material we can vibrate. It doesn't dampen the vibration. In fact, it seems to slightly amplify the vibration. And this is something we need to consider when we're doing our cell culture experiment. So this kind of raises the question of what are the cells actually experiencing? So if we look in terms of acceleration or peak acceleration, we can sort of calculate this based on the amplitude and the frequency of the vibration. And if we're looking at different frequencies of vibration in the lower range or in a slightly wider to 5 kilohertz range, we can calculate the accelerative level that the cells are experiencing. And we can see that at low frequencies, this is around sort of milli-G range. But as we get up to around 3000, 4000 hertz, we're getting into the sort of 1 to 10 Gs range. And obviously our accuracy of our device is a lot lower at these higher frequencies. So that carries through to this calculation. So in reality, the cells are experiencing forces that are several times larger than gravity at high frequency. And at low frequency, we're sort of modulating the gravitational force that cells are feeling. We've checked this in reality using AFM, atomic force microscopy, where we vibrated collagen gels using piezo actuators in the presence of an AFM cantilever with a bead, making force measurement of the collagen gel. Initially, we were looking for signs of strain hardening within the collagen material as the vibration was applying quite high frequencies to the material. But what we could see was that there wasn't really a change in the overall stiffness of the material so much as an introduction of this noise, which we realized was actually the 1 kilohertz vibration that we were applying. And the difference between A, B, C, and D is amplitude of vibration we're applying. So as we increased the vibration amplitude, you can see this noise increases linearly. So we can actually use AFM as a measure of the deflection, but also the force being applied to cells. And using this data, then we can sort of come up with a value of around 0.1 G for acceleration and forces in the nanonewton range being applied for each cell, depending on the size of the cell. So this ties in with the calculations from the previous slide. Moving on to why we want to vibrate cells. First, I'll just introduce what cells we've been focused on, and those are bone cells. And within bone, there is quite a dynamic environment involving a variety of cell types. Osteogenic cells or stem cells, which can form osteoblasts. These are cells which deposit calcium phosphate, the mineral component of bone. We also have osteoclasts that break down bone. And there's actually a balance between osteoblasts and osteoclasts, which allows your skeleton to remodel and develop throughout life and adapt to the mechanical loading that your skeleton exposed to. One example are astronauts. Obviously, there are problems, health problems with long-term space flight due to microgravity. And part of that is the loss of bone due to the lack of mechanical loading on the skeleton. So what's actually happening when an astronaut goes into space is the osteoblast activity is reduced and the osteoclast activity is increased, leading to a loss of bone density. So this balance is disturbed by the lack of mechanical loading. And we see similar things with osteoporosis on Earth, and also long-term bed rest, which has been used by European Space Agency to study space flight. So our interest was, could we increase osteoblast activity? Could we convert more stem cells into osteoblasts using vibration, and therefore have an impact on conditions like osteoporosis? There are a huge amount of factors which control this conversion from stem cells into bone cells, or even to other types of cell. Stem cells can actually become chondrocytes, cartilage cells, or fat cells, and there's some data suggesting they can even be converted into neuronal cells or muscular cells. We can use chemical methods like growth factors and hormones to stimulate stem cells. We can use other cells grown in co-culture. But what we've been very focused on are the mechanical factors that control stem cell development. Because we know that in the body, mechanical loading has such a big impact at the cell level. So just going back a little bit to 2007, our collaborators at Glasgow produced this paper where they grew stem cells on topographies of nanoscale size, and they found a very interesting result that if you take a square ordered array of nanopits and slightly offsets the nanopits by around 20 or 50 nanometers, in a random fashion, then the cells respond differently depending on the level of disorder. If we take a fully randomized pattern, then the cells respond differently again. And what we're seeing here are dense layers of mesenchymal stem cells from the bone marrow. And we can see that in red here, we have actin, but in green, we have either osteopontin or osteocalcin, which are markers of bone formation. And what we see is that these cells increase their bone formation on these disordered patterns. So even very small changes in the topography of 20, 50 nanometers have a drastic effect on how cells respond. And the conclusion was that cells exert tensional forces, and they're dictated by the availability of these nanopits which they can bind. So we can see significant increases in these bone markers in green within these two types of patterns. After 28 days, the cells even start to mineralize in the Petri dish, and we can see the formation of these bone nodules, but only in the case of the samples which have got this strong disordered array. And this ties into our work with vibration, where we find that over the course of 2 weeks, standard stem cells sort of retain their shape when grown on a static, non-vibrating surface. What we're seeing here is the cell nucleus in blue, and in white, we're looking at the cytoskeleton, the actin filaments of the cell, which exert the tension within the cell. However, when we vibrate at 500 hertz, we start to see significant changes in the shape of the cell. We see this significant spreading, and at higher frequencies, we see a more cuboidal morphology for the cells. And just looking at the microscopy, this does look more like a bone cell than a stem cell. And then we followed this up by looking at the gene and protein markers that the cells are expressing. I won't go into too much detail on the specifics of the markers, but here we have different genes related to bone formation, which are increased when we vibrate the cells for 8 days. And we have specific proteins such as osteopontin and osteocalcin, which again are increased with different frequencies of vibration. We also compared this to OSM, which is osteogenic media. So this is chemical stimulation of cell bone. And we're actually finding that the vibration in this case has a stronger response. If we look at whether the cells can form new bone, so new mineralized tissue, what we usually use to look at that would be stains such as von Coser and alizarin red staining. You can see that in black here, that the vibrated cells have more expression of this mineralized material. And also they change structure to form a sort of nodular pattern that we would expect for bone formation. If you compare that to the control cells, we see very low levels of mineralization, showing that the vibration is having a pretty drastic effect on bone formation within the Petri dish. We also looked at growing cells in collagen hydrogels. So in this case, the cells are not grown on a flat surface, but they're grown in 3D. And we see a variety of markers at different time points. These are all bone-related markers, and they're all increased in expression depending which time point you look at. We start to see that they return back to 0 after 21 days of vibration. We've also started looking at intermittent vibration to see if that has benefits in terms of keeping expression of genes for longer periods. Looking at mineralization again, we can see at 4 weeks and 6 weeks that the vibration group, 1000 Hertz, has higher mineralization at these time points. And OSM again is the chemical stimulation as a positive control. We can see that this is sort of equivalent to the vibration group. So I won't go into all the detail of the in vitro data we've collected, but in general, we find that the key factors that allow cells to detect these tiny vibrations include the adhesions of the cell to the surface or to the collagen gel using these integrins mentioned earlier, and also ion channels on the surface of the cells. Piezo channels are a type of channel, for example, that open when the cell membrane is stretched. So these are mechanically sensitive proteins on the cell surface, and when the cell is experiencing various forces, then we get calcium influx or various protein transporting the cell, and this can affect the cell function, change transcription, change how stem cells can differentiate and form other cell types, like bone cells. So a lot of our work recently has been focused on what can we do with these stem cells once we've turned them into bone cells. And I thought I'd briefly mention our translational work, aiming to use these cells as a bone graft material for bone repair. So what you can see on the right here is an example of a non-union fracture. And these occur in roughly 10% of fractures. So, but for 90% of people, when they break a bone, it will heal perfectly well, just given a bit of time and support. But in about 10% of cases, it might be more difficult, and the fracture will not necessarily heal on its own. Or it might be the case that certain fractures are too large to heal on their own. For example, if you have a bone cancer and there's a large section of bone removed, then there'll be a need for graft material to replace the missing section of bone. Fractures can also occur due to osteoporosis, and this specifically is a lack of osteoblast activity, so a lack of bone formation, leading to low bone density and a higher risk of fracture. The cost of non-union fractures is very high. Each case can be tens of thousands of dollars. And due to the amount of cases worldwide, we're looking at billions of dollars of healthcare expense. And sometimes there is a need for materials to help repair these defects in the bone. So that's where our stem cells that we've vibrated come in. At the moment, we're testing a mixture of these nano-vibrated stem cells mixed with a ceramic granular graft material to give structure. We're using the ceramic material as a carrier for the cells to hold them in place once we implant them. And our plan is to implant them in a variety of different bone defects, starting with hand fractures. But eventually, we're aiming to look at these sort of significant tibial defects that you might see in the lower leg, using the cells and the granules as a filler material. This sort of relates to a project we've had ongoing with the Sir Bobby Charlton Foundation, which is a charity focused on landmine survivors. And in some of these cases, the difference between retaining a leg after a landmine injury can be as a matter of providing a good graft material which could fill a defect of significant size. Otherwise, we find that a lot of the survivors of this type of injury end up losing limbs, and it has a knock-on effect, obviously, for their quality of life and their capability to work, for example. So at the moment, we're working with a GMP cell manufacturer to produce nanokick stem cells. So we've taken our devices and implanted it into their production process. So they're taking stem cells, mesenchymal stem cells from fat, adipose tissue. They create a cell bank of these cells. And then over a period of 4 weeks, they vibrate them and they produce several batches of cells for us to test. And in the end, we're aiming to combine this with the granular graft material in surgery and implant that into a bone defect. At the moment, we've been very focused on collecting safety data and efficacy data with these stem cells, getting the manufacturing up and running with Histocell. It's a non-trivial task to take an academic technology into a GMP environment. The clean rooms that they use are very high grade. Obviously, they can't afford to have any pathogens end up in the final product. It's a 2-year process to take technology from the lab basically into their clean room environment. We're also focused on trying to increase the number of cells that we can nanovibrate. I mentioned that a Petri dish only holds a few thousand cells. For each patient, we're looking at delivering around 20 billion cells. So you can imagine that be very careful how we're applying our vibration stimulus to these large cell culture vessels. We're also developing a protocol for human trial involving hand fracture and working with the regulatory agencies, the UK, the MHRA, aiming to get approval for this human trial. So I mentioned scale up, just to give you an idea of what we're doing at Strathclyde. Our devices previously were capable of stimulating flasks of a few million cells. Then we've recently validated the use of these large multi-layer cell stacks for growing cells in monolayer on multiple levels. We can get around 50 million cells at most per batch using this. This is enough to treat a couple of patients, really, or maybe only one patient, depending on the amount of quality control required. So obviously this has its limitations if we want to treat many patients. And so with colleagues at Strathclyde, we've been looking at more advanced cell culture systems, such as these hollow fiber cartridges, where we're able to grow hundreds of billions of cells, even a billion cells. And at the moment, we're trying to validate our ability to vibrate these hollow fiber cartridges and ensure that the cells grown inside are actually receiving the 30 nanometers of vibration that we know is good for bone formation. So we've done a little bit of modeling and also a little bit of scanning laser interferometry showing that we can vibrate these large scale culture vessels. This is the 5-layer cell stack. Interestingly, we've lost the sort of piston motion of the vibration, and we are seeing these nodal patterns form on the top surface of the cell stack. But reassuringly, the amplitude at any given point is pretty much the 30 nanometers we're expecting. We just might have the cells receiving this out of phase from each other, depending which part of the surface they're being grown on. Our first human trial, sorry for the gruesome image, is planned to be a set of hand fractures. We're working with a local clinician, Professor Andrew Hart, who will recruit 6 patients locally in need of graft material of around 3 cubic centimeters. And we'll use our granules and cells as a graft material. And this 6-patient study will be a safety study, aiming to check that the stem cells are safe for human use before moving on to larger bone defects, where we're looking for signs of efficacy. So just to conclude, I thought I'd mention our future questions that sort of arise from this research. We know that cells are responsive to vibration, and we know that they can form bone just using vibration as a stimulus. But we're also interested in looking at other stem cell types, other cell types. For instance, can we form cartilage? Could we form neuronal tissue using vibration? And we're looking at a variety of other cell types, including bacterial biofilms, and also other cell effects such as cell adhesion, cell growth, and can we modify these using vibration? One way to do that might be to alter the amplitude and frequency of the vibration. We still don't understand how responsive cells are. We have recently published data suggesting higher amplitudes could be beneficial. And we might find that other frequencies, lower frequencies, are better at getting other cell phenotypes. Additionally, Stuart Reed has been working on an STFC project aimed at trying to use vibration on patients directly. So rather than taking stem cells, growing them and vibrating them, can we directly stimulate the stem cells within people's bone marrow using wearable devices, which is the one you can see here, aiming to deliver this sort of stimulation directly into the bone, where it might have benefits for conditions such as osteoporosis, helping to restore the balance between bone formation and bone resorption. So I think that's my time up. I'd just like to thank everyone at Strathclyde and our collaborators at Glasgow and all the previous people who have worked on this research. For the data in this project, particularly Paul Campsey, who's done a lot of the device development and also the scanning laser vibrometry, and obviously our funders, UKRI and the EU, and the Sir Bobby Charlton Foundation. So thank you.
Thank you very much, Peter. That was quite impressive. I think I'll see if there's anyone on the participants that have any questions. So if there's any questions, just please feel free to ask.
Yeah, Pete, nice talk, thank you. I have a question, and it may be because I missed part of your talk because I had to step away from time to time, sorry. But from what I understood, it looks like you have to apply a wide range of frequencies in the vibration and not just one particular frequency. Is that right?
Yes, we aren't entirely sure how specific the cells are to the frequency and amplitude. We do see mineralization at 1000 hertz. We see it at 3,000 and 5,000 hertz. We see less of it at 500 hertz. So there might be a point, a sort of plateau point, where we're stimulating the cells sufficiently using certain frequencies. We've picked 1000 hertz for our experiments, partly because the device we know is consistent. If we go to those higher frequencies, the device is less consistent. So that's part of the choice. In terms of the amplitude, we're developing new electronics to allow us to go to higher amplitudes, but we have compared 30 and 90 nanometers, and 90 nanometers does seem to have a slightly better cell response. So it might be that higher forces on the cell could give better response. The other thing to consider is we're culturing the cells and vibrating them for 28 days solid, so they're not getting a break. And we might find that the cells respond better by breaking up that stimulation, allowing them to return, relax mechanically, and then stimulating them again. There's lots of things that we can look at, and lots of project work ongoing in that area.
Okay, so when it comes down to actually stimulating the cells, you just apply 1 kilohertz vibration.
Yes,
Okay.
we have tried different frequencies side by side, but obviously it's a huge parameter space, and if we're doing 4-week experiments, there's a limit to how many frequencies we can look at a given time. Originally, there were experiments at low frequencies, 1 and 10 hertz. Adam Curtis started with physiological frequencies, and he did see some effects with endothelial cells, although when it came to stem cells, they didn't respond to those low frequencies.
So sorry for hogging the questions, but when you mean physiological scales, you mean like wavelengths that correspond to the cell sizes approximately? Is that what you mean?
I'm meaning more the heartbeat or walking speed or respiration is all around 1 to 5 hertz.
Sure. Okay.
That's where the original thought on Adam Curtis's part was, can we use sort of physiological rates of mechanical loading.
Okay.
For stem cells, it seemed that the overall magnitude would be a lot higher.
Okay. And so is the philosophy basically you're just trying to replicate the vibration? So at the very beginning, you had this video with the cells just sort of naturally, I guess, vibrating or something like that. So the philosophy is to try and replicate that or enhance that or something like that. So you want frequencies similar to that, or is that not the right way to think about it?
That was the original thought, and that was Adam Curtis's idea. These membrane motions were around the sort of 1 to 10 hertz sort of region. Can we play that back to the cell using the surface motion? But we're finding that different cells respond at different levels. So there was a PhD student, Habib Nikikar, who worked with Adam during his PhD. He started at these low frequencies with stem cells, and they didn't respond. So he decided to crank it up to 1000 hertz and higher, and they started responding in terms of mineralization, these markers of bone formation, which suggests that the cells actually, depending what you're trying to get the cells to do, will require different levels of force being applied to get them to respond. We've also looked at bacteria, and we found that actually slightly lower frequencies give some response around the 100 to 1000 hertz range. And one of my colleagues, Sean, carried out quite a methodical study looking at different frequencies, and there was sort of ramping up of effect above 100 hertz. So originally the plan was to look at physiological rates, but quickly became apparent that you go higher in frequency, you get a bit more response, typically.
Okay, thank you.
Are there any more questions for Peter? Okay. Maybe while people think of questions, I have a few or a couple, depending on how much time between people not asking questions. So my first question really is like a two-part question. So the first sort of beginning bit is, at what stage are the clinical trials? So obviously I'm slightly aware, but it would be nice to share that. And a sort of side to that. So I know this project is a mix of subjects. You come in from physics and astronomy, sure it's the same, but there's also like bio people, and now you're on the biomedical department. So there's clearly a very mixed range of expertise in there. But at what stage do people that, like yourself, that do physics and astronomy still get involved in the clinical trials, or do they just do the experimental part, and once it gets to the clinical part of it, does it just stop?
Yeah, it's a good question. I'll just point out actually that at Strathclyde we have 6 people working on nanokicking, and all of them have an undergrad in physics. We do work with everyone at Glasgow who are definitely molecular and cell biologists, but some of us at Strathclyde are doing that sort of work too, or bits of it, cell culture and so. In terms of the clinical trial, at the moment we're still getting the manufacturing sorted. Obviously we need these cells to be produced to a clinical grade before they can be used on humans, and we expect that to be the first batches to be produced before Christmas. We also need to collect safety and efficacy data. The efficacy data in various animal models is already underway, and the safety data will be carried out next year, we expect. And that will involve checking for tumor formation. Do the cells end up somewhere bad? They don't end up in the brain or anything like that. And that has to be carried out before we're allowed to put these into human. And so we're sort of expecting by the end of 2022, mid 2023, we'll be hopefully in a position to apply to do a human trial. It's been, a 10-year road from the initial experiments to get to this point. So you can imagine that for more complicated products that aren't just stem cells and some granules, that it could be even longer before they get to clinic. But yeah, we're aiming for the next couple of years to try and get to that point. We're making progress towards that. It's certainly, it takes a lot of effort. Our team working on it is roughly about 20 people now, maybe 30 even, across several universities. You asked about people entering into the sort of clinical side of things. I would say having some experience in wet lab work, the cell culture itself, I got that during my PhD. But actually, that's helped me to sort of understand what's needed for data-wise in terms of the regulatory submission for clinical trial. And I wouldn't say I'm directly involved with delivering a clinical trial. We have clinicians who will do that, but we do need to be part of the planning for that, and making sure that we have all the preclinical data to give to the MHRA to allow us to do the trial. So I would say definitely physicists can get involved with the planning of the trial, but it's always going to be the clinicians that deliver it, and the NHS in this case.
Okay, and if anyone has any questions, just please raise your hand and or start speaking. Otherwise, I'll ask 2 more questions that have sort of follow up from that. So from what you're saying then, from your own experience then, as a physicist that then went into biomedical, is there any advice that you would give people from like, do you somehow see that because you did your undergrad in physics and astronomy and then did your PhD on this, did you feel like you missed out on anything or any sort of extra education that you could have done in between? Or was it a very natural sort of from one side into the bio side of things? And not really related to that, so you were talking about animal trials and stuff. So from other stuff, when it comes to medicine and stuff, they've kind of been showing that the actual correlation for medicine at least, so I don't know how it works on this, but in medicine stuff, the correlation between what vaccines and other medication that you can give animals when they're doing the trials to the actual passing to humans, there doesn't seem to be any benefit. So because animals actually react very differently to humans. So do you expect to see something like that in this case, or because I guess you're not really injecting them with any chemical sort of stuff, is a different mechanism?
Yep. Both very good questions. So the first point on career sort of changes. I made the switch at the start of my PhD. What I would say was that was a very tough learning curve because I'd never done biology in high school. I didn't do it during my undergrad degree. I was coming into that completely cold, and I managed to pick up the ability to grow cells in a wet lab. I think it's actually easier for physicists to pick up cell culture than it might be for a cell biologist to pick up electrical design and some of the physics bits and pieces that we do. I think, you know, cell culture is like cooking. It's following a recipe usually. Whereas I would say with physics coming the other way, there's a lot more design work, which might be harder to pick up without having some background knowledge in it. So I actually think physicists going into cell biology is the easier way to go around it. The other question, I fully agree that animal models do not represent human models very human models. Animal models don't represent humans very well at all, and there's a huge attrition of drugs and things that work in animals and then don't work in humans. I think something like, I'm not sure if it's around 90% of the level of things that go into human trial fail, and that's why it costs $1 billion for a single drug, because so many have failed along the way to get to the one that does work. So I fully agree that it doesn't really give us strong evidence that anything's going to work. On the safety, it probably does minimize the risk of people dying in early clinical trials. And that's why the MHRA and the EMA are so stringent on, you have to have this data, otherwise you shall not pass. You won't get into human trials without that data. But there's a huge effort to try and reduce animal usage, actually. At Glasgow University, they have a whole CDT, Centre for Doctoral Training, focused on non-animal technologies, trying to develop in vitro cell culture-based methods of, detecting safety in products and efficacy. I think that is going to grow, and there's a huge focus on trying to obviously reduce animal usage and eliminate it as well. So getting alternatives is a big focus for a lot of researchers now, because it doesn't work very well. It is really not a good representation of what happens in a human.